Okay, this is part 3 of Mitochondrial Insights. I recently gave 2 lectures, parts 1 and part 2 of Mitochondrial Insights. The main thing to focus on today, we're going to start going into, talking about succinate. I'm going to give a subsequent letter on succinate because succinate is a big subject, but we'll just get introduced to succinate. A very quick overview of the mitochondria. It's thought to be endosymbiosis, a bacteria that was aerobic originally entered into an anaerobic bacteria. Here's the outer mitochondrial membrane, OMM, intramembranous space, IMS, inner mitochondrial membrane, IMM, and then the mitochondrial matrix, which is all in yellow. The inner mitochondrial membrane is the green, okay? The mitochondria is basically like a minor miracle. It's a nuclear power plant that splits the atom, it splits the hydrogens, and these are originally removed from glucose. And the proton component from the nucleus is pumped into the intramembranous space, that's H+. The electrons are passed down a gradient of electron grabbers called protein complexes that have progressively higher electronegativity, desire to grab electrons. Oxygen is the ultimate electron acceptor at the end of the electron transport chain. Hydrogens get added to it and it's converted to H2O, to water. Okay, what you've done here is build up a proton gradient called a proton power gradient. It's like having compressorized air, and then the gradient is harvested, like releasing the compressed air into the mitochondrial matrix again. And that energy spins ATP synthase here, which is also called complex 5, and that enables adding a phosphate to ADP, adenosine diphosphate, to become adenosine triphosphate with 3-phosphate. This is just a basic overview of mitochondria. I'm going to come back to this guy right here, complex 2, because that's got the enzyme that we care about, succinate dehydrogenase, and that's going to be the key central point of the mitochondria. Okay, I can remember when I was a medical student, there was a book about EKGs. I think it was called Rapid Interpretation of EKGs. The author's name was Dale Dubin. It was a very clever book, and it said that if man is the center of the universe, and the heart is the center of man, and the sinoatrial node where the heartbeat begins is the center of the heart, then the sinoatrial node is the center of the universe. Okay? And what I'm getting at here is we're going to go deeper. If the center of the sinoatrial node is its mitochondria, and the center of the mitochondria is succinate dehydrogenase enzyme, then succinate dehydrogenase enzyme is the center of life on Earth for humans as we know it, okay? And this right here is the center of the mitochondria, because complex 2, succinate dehydrogenase enzyme, it connects Krebs cycle to the inner mitochondrial membrane, and that's going to end up being very important. It is the tipping point of the mitochondria. It is the valve between oxidative metabolism and anaerobic metabolism. It's also the blow-off center. Like when there's a shortage of oxygen, glycolysis, instead of heading into pyruvate and acetyl-CoA and going into the mitochondria with that as acetyl-CoA, it can just go into lactic acid, go into lactate. That's an option. And what the mitochondria can do is when it's having problems, it can just boot succinate out of the mitochondria. This will all make more sense in a moment, but what I'm letting you know, succinate, complex 2, succinate dehydrogenase is the enzyme, succinate as a substrate, is really the tipping point of mitochondria. So here's another drawing of mitochondria, just another way to show it. And again, these are the electron carriers. Electrons are passed down. Okay, then the protons are pumped out. Here's complex 2. So complex 1 is intimately associated with the cofactor NADH, okay, and that delivers its electrons, gets made into NAD+. FADH2 delivers its electrons, and it gets made into FAD, okay. Then coenzyme Q carries electrons. Complex 3 also pumps out a proton, okay, and that's complex 4, also pumps out a proton, and that's complex 5, which is ATP synthase, makes ATPs, okay. But this is what we're going to be coming back to, complex 2. Okay, we talked about it before, how saturated fat in particular of the fats, more so than the other fats, inhibits intermitochondrial membrane electron transport in this region here around coenzyme Q and complex 3. And then you get a backup of things, including you get a backup of succinate dehydrogenase, this enzyme here, complex 2. Okay, then succinate, the substrate for this, is going to accumulate, and that's going to have major consequences for the cell. Besides causing insulin resistance, it's going to cause some other things too that you don't want to have happen. This is just showing what happens when you have block here, like let's say due to SAT-FAP. You'll get electrons can no longer be transported along here, so they'll start to drop down into the matrix. They'll combine with oxygen to make superoxides. That's a free radical with the extra electrons unpaired in its outer orbital. And it's usually quickly neutralized by superoxide dismutase, SOD. Gets made into hydrogen peroxide, and that's converted into water. It's usually not a big deal, but if it's excessive, there's too much of it, then it can overwhelm these neutralization enzymes and cause damage to the mitochondria, the inner mitochondrial membrane, or even the DNA in the mitochondria, for example. Okay, so the cell does not want that to happen. Okay, here's just showing the connection between the two cycles. So here is Krebs cycle, which normally in humans moves in a clockwise fashion. Here is the substrate, succinate. Succinate is then typically, by succinate dehydrogenase, it's converted into fumarate, where it basically just gets a double bond, okay? And then it gives off its electrons, and they convert FAD to FADH2. Those electrons are passed on to ubiquinone to become ubiquinol, okay? And this is how you connect Krebs cycle very directly to the inner mitochondrial membrane and electron transport. If you have injury to succinate dehydrogenase enzyme because it's inhibited for some reason, or if just the whole cycle is backing up, especially near this vicinity, succinate will accumulate. You can have mutations of succinate dehydrogenase enzyme, and those are associated with some rare tumors, some paragangliomas, gastrointestinal stromal tumors, things like that. Okay, but the key thing we got to think of is that succinate is going to accumulate, and when it accumulates, it has a way to exit out of the mitochondria. That's sort of like an exit valve for accumulation. This is just showing it in a little more detail, the enzyme, and it's got different subunits. Succinate going to fumarate, giving its electrons to FAD to FADH2. They're passed through a couple iron sulfur clusters. Then from ubiquinone to ubiquinol. Okay, so that's going to have a ketone on it, a carbonyl group, whereas this is going to be reduced, the alcohol group instead of a carbonyl group. All right, and this is the connection between the tricarboxylic acid cycle, Krebs cycle, and electron transport chain. It's also unique in the sense that it's the only one of the inner mitochondrial membrane complexes that is entirely, all of its DNA comes from the nucleus. None comes from the mitochondria itself. It's the connecting point. It's going to be the blow-off point. One of the points I'm going to make, though, is this could also lead to a vicious cycle. When you see the subsequent events that are going to happen with the accumulated succinate, it'll make more sense. I'll show you just in a moment what's going to happen. We talked recently in one of my lectures about hypoxia inducible factor. When you have a lack of oxygen available, normally with normal oxygen, hypoxia inducible factor, HIF alpha, will get hydroxylated. There's a prolyl hydroxylase domain enzyme which hydroxylates it. You need vitamin C for that as well. And then the hypoxia inducible factor gets ubiquinated, which is like, eat me stickers put on it, and it gets chopped up in the proteasome, which is like a wood chipper. So that's what happens in normoxia. The cell is constantly making HIF, hypoxia inducible factor, but destroying it. Okay, so that's a strange thing to do. Why would a cell keep making a protein and destroying it? Because it needs a good sensor. It's like an alarm system for when hypoxia occurs, because if the hypoxia is prolonged, the cell will die. So it's a way to have a quick response to hypoxia, so hopefully it won't be killed by hypoxia. And in our ancient ancestors, they didn't have a lot of atherosclerosis. What they had was a lot of infections. So that can cause a lot of inflammation, leading to tissue hypoxia. In the setting of tissue hypoxia, hypoxia-inducible factor alpha, there's no oxygen available, not an adequate amount of oxygen available to hydroxylate it. Okay, so prolyl hydroxylase domain enzyme, PHD, can't hydroxylate it, in which case the HIF alpha will go to the nucleus, it'll form a dimer with hydroxy-inducible factor beta, bind to HRE, hypoxia response element, and then transcribe a bunch of genes to respond to hypoxia. In particular, something called VEGF, vascular endothelial growth factor, to stimulate angiogenesis, the ingrowth of blood vessels that will hopefully be made in time to get more oxygen to the tissue. Okay. It'll also upregulate, meaning increase enzymes for glycolysis, so you can generate energy anaerobically. And this all leads to a type of metabolism in a cell, largely anaerobic, that's very similar to cancer. Okay, so hypoxia induces changes in the genes that are expressed in a cell, gene transcription, similar to that of cancer. So that's another reason why you don't want to be inducing hypoxia. What's the big things that induce hypoxia in the diet? High-fat diets and high-sodium diets. Okay, so not a good idea. And then it's made worse if somebody's smoking cigarettes. And it's also going to be interesting that when succinate accumulates, succinate will also, it'll diffuse out of the mitochondria, it'll go into the cytoplasm, and it also will inhibit prolyl hydroxylase domain enzyme. So it'll also block this step to contribute towards the HIF alpha not being destroyed and thus going into the nucleus and transcribing for the genes to respond to hypoxia. Okay? And like I said too, you can get succinate accumulating in the setting of hypoxia. It'll accumulate in the setting of something that's inhibiting succinate dehydrogenase enzyme. That's called pseudohypoxia. And then there's rare cancers that are associated with mutations in it. It's also the case when you get the bacterial endotoxin LPS binding to the toll-like receptor number 4 on macrophages, that this will induce, it'll cause elevated succinate, and you'll get the same response. So inflammation has a very similar response as hypoxia, has a similar response as cancer. And this brings home the point that cancer is associated with hypoxia and inflammation. And it's like the idea of, the old disease of hot disease, cold disease. Hot disease is inflammation and active infection, for example, or injury, healing with fibrosis, leaving scar tissue, making the tissue farther in distance from the blood vessel oxygen delivery, and can cause some chronic hypoxia, which can potentially lead to cancer. So it's like the idea of cirrhosis. You can progress from fatty liver to NASH, inflamed fatty liver to cirrhosis, scarring, fibrotic tissue, and that sort of bordering off normal tissue, making it hypoxia, which can lead to liver cancer, hepatocellular cancer. So this is a mechanism by which inflammation anywhere, Barrett's esophagus from gastroesophageal reflux, scarring, fibrosis, metaplasia of the liver, of the esophagus, scarring it down, causing some normal cells to be trapped in there that can potentially transform into cancer, like by a Warburg effect, become hypoxic, and thus this compensatory mechanism of shifting from being a worker cell, well oxygenated, making a lot of ATP, to becoming like an anaerobic bacteria, just wanting to proliferate and running primarily on anaerobic glycolysis. Okay, so I'm going to show a couple more things on succinate because it's quite interesting. And remember, this is just the introductory talk to succinate. I'm going to go into more detail next time around. Okay, so here is a paper, Cellular Succinate Metabolism and Signaling and Inflammation, Implications for Treatment, by Zhang Kai. There's been a couple good papers by some of these Asian groups. Okay, so I just wanted to show, how does the cell deal with succinate when it's elevated in the context of hypoxia, okay? So you've got a blockage of succinate dehydrogenase enzyme due to, let's say, hypoxia. And the hypoxia, again, could be due to, in our ancestors, it was most often due to infection. In our modern people, hypoxia is often due to chronic atherosclerosis because of hypertension and diabetes. Also, acute hypoxia related to high-fat diet, sticking the red blood cells together, blood sludge, rouleau formation, and sodium, vasoconstricting, inhibit endothelial nitric oxide synthase. So those two big things drop in it. And then it gets worse because typical person's eating these high-fat diets, so they're getting some bacterial endotoxin across their gut border, and that's causing amyloid clotting, which is going to make the blood thicker, cause increased tissue hypoxia. And those bacterial endotoxins getting across LPS from gram-negative bacteria, LTA from gram-positive bacteria, they're also going to be binding to some of these immune system cells, like these macrophages, like these, if they get across the endothelium, these dendritic cells, residential macrophages, also called sentinel macrophages, and then the LPS bacterial endotoxin, for example, activating the toll-like receptor number 4 in the macrophages, and then that causing succinate accumulation. Okay, now why would a cell want to cause succinate accumulation? And here's the reason. Just like lactate is the blow-off valve for anaerobic conditions for glycolysis, succinate is the blow-off valve for the mitochondria for Krebs cycle. And it also becomes like Paul Revere. It's going to jump out of the mitochondria and start activating things in other locations. So here's what I'm talking about. Succinate dehydrogenase runs into hypoxia. It starts to go in reverse, okay, which can convert fumarate to succinate. Succinate accumulates. It'll exit the mitochondria through this, what is called DIC, but it's really DICC, dicarboxylic, because it's a dicarboxylic acid, succinate, its chemical structure. Fumarate and succinate are sort of like a dicarboxylic version of butanoic acid. So it's like a dioic butanoic acid, okay? Fumarate is just like only difference is it has a double bond in its center. Anyway, succinate accumulates. It'll exit through the dicarboxylic carrier because it's a dicarboxylic acid, DIC, and then that's to exit the inner mitochondrial membrane. Then to exit the outer mitochondrial membrane, it goes through something called VDAC. You don't need to know all this. What you need to know is that succinate can exit the mitochondria. I just find it a little bit interesting to know the carriers because I deal with these carriers when I'm reading about other things too. Voltage-dependent anion channel VDAC on the outer mitochondrial membrane. And by the way, this thing is a key site related to mutations of cancer. We're going to come back to that later. Hexokinase 2 is going to attach to VDAC in the setting of cancer. I talked about that in my cancer lectures, cancer biochemistry lectures. Okay, so succinate now has exited the inner mitochondrial membrane. It's now exited the outer mitochondrial membrane. It travels to the plasma membrane, and it exits the cell through something called organic anion transporter, OAT. Then it will diffuse into the extracellular space, and it'll bind on receptors of nearby cells. The relevance is that by succinate exiting the mitochondria, it not only goes into the cytoplasm, but it exits the cell to the extracellular space, and it communicates with adjacent cells. So it's a way that an intracellular problem in one cell of hypoxia is communicated to neighboring cells, which are also likely to soon, if not already, experience hypoxia. And it's basically like Paul Revere sending the alarm bells. Hypoxia is coming, which usually meant infection, inflammation is coming, okay? So what it's trying to do is get these cells. They don't want to die. They're going to die if the hypoxia gets worse. So by calling for angiogenesis, they're trying to prevent the infection that they believe is making them hypoxic and to start growing new blood vessels, angiogenesis. But think about it. That's what cancer wants, to grow new blood vessels, to get more nutrients to it. So the response to hypoxia is basically very much like the response of cancer trying to get itself to grow. So here's Paul Revere, alerting everybody, 1 if by water, 2 if by sea. Okay, so that's what succinate is doing. It's taking an intracellular problem and communicating it to its neighbors to warn them. Okay, so here's a paper about succinate. Succinate metabolism related to inflammation, ischemia. Okay, it does a few things. It's like the key point. We're not going to go into all this, but I just want to show you a couple things about succinate. So when succinate accumulates, it activates immune cells, and they also activate it when they're activated. So it's a back and forth communication because it's functioning as a signal message. Okay. So again, if LPS bacterial endotoxin binds a macrophage, it'll then cause accumulation of succinate, and that can interact with other cells. If you've got hypoxia inside a cell, that will increase succinate because it'll inhibit succinate dehydrogenase enzyme. So succinate is a substrate. Because the enzyme's not working, the substrate will then accumulate. All right. You can get that also with mutations of succinate dehydrogenase enzyme, which are rare. They're associated with some rare tumors, things like paragangliomas, which are really rare tumors. I hardly ever see them, but I do see them sometimes. Okay. Now, if you're taking this succinate out of the mitochondria and it's going and traveling and going off to alert nearby cells, then you're depleting the Krebs cycle intermediates. But, in the setting of high protein availability, the glutamine, it just gets converted. The glutamine gets converted to alpha-ketoglutarate. Glutamine, you just deaminate it. It's just the same thing like as alpha-ketoglutarate. So you can keep the Krebs cycle going. This idea of replenishing Krebs cycle intermediates, that's called anaplerosis. You'll hear that term sometimes. Okay, so that's how succinate can go to the cytoplasm, but the Krebs cycle intermediates are not depleted. Okay, this trend of immune system cells switching to anaerobic metabolism is basic to immune system cells. It's called a metabolic shift, when the immune system cells shift from oxidative metabolism to anaerobic metabolism. The way you can think of it is the immune systems are kind of like a bunch of firemen. A lot of times they're just sitting around at the fireman's lodge and they're having a good time. They're cooking a different type of meal. They're joking around with each other. Everything is cool. So when oxygen is widely available and there's no infection to fight, they sit around having a good time. Everything is cool. Hey, how you doing? Telling jokes, cooking food, having a good time. But when there's a fire, they got to respond to it. Go, go, go. And they have to run an anaerobic metabolism because in the center of an infection, there's not going to be hardly any oxygen available. Okay, so they have to run anaerobically. So that's a key point. That's the metabolic shift from oxidative metabolism to anaerobic metabolism. And the relevance of this is that every cell in the human body has the same DNA. It's just a question of which genes are activated. So the point I'm making here is this makes it obvious that the metabolic theory of cancer is totally feasible. Any cell theoretically could shift on these genes of primarily anaerobic metabolism when it needed to. And okay, so it totally makes sense. Everything fits with it. And like I had mentioned, it's not changing one gene, it's changing a whole bunch of genes to function anaerobically, to change from being a worker mentality. Working with oxygen is mostly a worker mentality for most cells, versus for most cells to go hypoxic is to become like an anaerobic bacteria. The immune system cells are uniquely adapted to do so because they have to function in hypoxic environments, okay? Now, when the individual cell became hypoxic that we started out talking about, succinate accumulated, became the signaling molecule, okay, and it'll also upregulate its glycolysis enzymes because it's preparing itself to function with low oxygen. And that's one of the things that HIF did when it became a dimer and activated gene transcription of hypoxia response element on the DNA to increase angiogenesis. It also increased glycolytic enzymes, okay? And it's also mimicking cancer tumor physiology, okay? And then this is also showing not just the macrophages, but also the dendritic cells. Those are, residential macrophages, or sentinel cells are also kind of called. So what I'm trying to say is this is how the cell shifts back and forth from aerobic to anaerobic metabolism, with succinate being a pivot point, okay? And I was also making the point that if a cancer cell had to do something like this, glutamine will just replenish everything. Glutamine's got the nitrogen on there, so you can take that nitrogen, you can make that into proteins, you can take that nitrogen, you can run it into the pentose phosphate pathway, and you can make your nucleic acids. So you can run everything off of glutamine. You don't need glucose at all. But you need glucose in the human body for the brain, you need glucose in the human body for the red blood cells. Okay, so what I'm trying to say is glutamine is what I would worry about, not glucose, because a lot of people are obsessed with this idea of sugar feeds cancer. No, no, no. Glutamine is the dangerous thing because sugar doesn't have any, glucose doesn't have any nitrogen. Glutamine does. Okay. Okay, so I just wanted to make the point. Tissue hypoxia, as you routinely get, our ancestors got it from infection, but modern human, they get it from a high-fat diet, causing blood sludge, rouleau formation. They get it from high sodium, causing vasoconstriction, narrowing of the arteries, inhibition of nitric oxide, endothelial nitric oxide synthase. And number 3, they get it from leaky gut, the LPS causing amyloid clotting. All right. They also get it from elevated uric acid, like too much high fructose corn syrup, for example, too much meat. With the purines, you'll get that elevated uric acid. That's also a vasoconstrictor. So that's all bad. But that's why I like my diet, low sodium, plant oil. And then the other thing they get it from is they get it from medications. And so you'll have tissue hypoxia from those things, that type of meal, and also from baseline accumulation of atherosclerosis related to hypertension, high cholesterol, and diabetes. And then tons and tons of common medicines. They're mitochondrial inhibitors, so you get that from the sat fat, we talked about it. But atrazine also inhibits complex 3, so that could cause an increase in succinate. Cadmium, which is a common thing in processed foods especially, can do it. Okay, we got lots of mitochondrial inhibitors. the F minus that's in water. Further down the path, you get omega-6 coconut oils leading hydroxynonenal inhibiting ATP synthase. Okay, statins are right in this spot, inhibiting coenzyme Q. Not good. Same with Plavix, amitriptyline, the antidepressant. Not good. Antifungals are inhibiting complex 2. That's a freaking disaster. That's processed food. What I'm trying to tell you is, if you understand what I'm saying here, processed food is a disaster. Number one, the antifungals inhibit complex 2. Okay, that's what we just talked about, something inhibiting complex 2. Complex 2 means succinate dehydrogenase, the key enzyme. So they're inhibiting right at the tipping point, the pivot point, the blow-off point for the mitochondria. That's bad. So, yeah, oh, fine, it's a good preservative for preventing mold growth in food, but it's also damaging to our cells. That's really bad. That's a big baddie. Okay, and this is the FADH, intimate electron carrier. Complex 1's intimate with NADH, okay, and that's inhibited by a lot of stuff. And you're going to know these medicines: metformin, selective serotonin reuptake inhibitors, PCBs and unfiltered water, Lasix, a common diuretic, arachidonic acid, common in meat. That's the bad omega-6, pro-inflammatory. Chromium, amiodarone, mercury. It's bad. Haldol, antipsychotic medication, trichloroethylene, like from dry cleaner stuff, liquid paper, degreaser cleaners. Okay, this stuff's all bad. So anyways, also Tylenol is a complex 3 inhibitor. That's a bad thing, all right? I just wanted to give you a sense of what's going on. So we're going to come back to succinate in the future lecture and go into more detail about it, but I'm letting you know it's a key tipping point between going into hypoxia pathways versus normoxia pathways. It's quite interesting. So I hope you found that interesting. And that's it for part 3 of Mitochondrial Insight.